So hello everyone, I am Michelle Ma, one of the technologists here at the Advanced Molecular Diagnostics Lab located in the Princess Margaret Cancer Centre, part of the University Health Network in Toronto, Canada. For those of you who are not AGT members, please consider joining the organization so that AGT can continue to be the voice of the technologists in the community and be able to offer programs like this in the future. Please check AGT's website, www.agt-info.org, and to see the many benefits of the membership. I've been a member since I was a student, and I've really appreciated the variety and also the constant inclusion of more materials that pertain to molecular genetics. So please do consider. All right, a few housekeeping items. Feel free to ask questions during the session. In the upper right side of your computer screen, you will see a control panel. In the lower portion of that panel, you can type in a question or comment and submit it to the webinar organizers. You can do this at any time during the presentation. We will reserve 10 minutes at the end of the presentation for Q&A. If you are participating as a group, please designate one person in the group to complete the online evaluation. In order for everyone to receive their certificates of attendance, that person will need to enter the names and emails of each person who is participating at your site today into the evaluation. As a backup measure, a sign-in form can be completed in fax or email to AGT as well, and the certificates will be emailed approximately 30 days following the webinar. So now, to begin, it's a privilege to introduce my Associate Director here in the lab, Dr. Tracy Stockley, who will discuss the detection of EGFR mutations in cell-free DNA from non-small cell lung cancer patients using the Oncomine Lung cfDNA assay. Dr. Stockley is the deputy head here of the genome diagnostics at University Health Network, UHN, and this includes oversight of the Advanced Molecular Diagnostics Lab at the Princess Margaret Cancer Centre, as well as the Genome Diagnostics Lab located right across the street in Toronto General Hospital. Dr. Stockley is also an associate professor in the Department of Laboratory Medicine and Pathobiology at the University of Toronto. Her role at UHN includes oversight of genetic testing for oncology, including companion diagnostics, genomic test development for clinical laboratory use, and applications of next-generation sequencing to clinical care. So welcome, Dr. Stockley.
Great, thank you very much, Michelle, and it's my pleasure to be here and speak with all of you today about the work we're doing here in Toronto. So as Michelle mentioned, I'm going to speak today about how we've recently set up to look at mutations in cell-free DNA from non-small cell lung cancer, and in particular using a next-generation sequencing assay called the Oncomine Lung assay. And just so that you know, in case you're not familiar with our labs, as Michelle mentioned, we do have two labs here in Toronto. Our lab at Toronto General Hospital is what we consider as our high throughput routine lab, where we do many assays for inherited and acquired cancers. And then across the street at Princess Margaret Cancer Centre, our lab there is more of a translational lab where we look at new assays and work with our oncologist colleagues on trials and new projects. And so this work is something that we've really done more at the Princess Margaret Centre, where Michelle is also working. Okay, so just to begin then, I just wanted to give you an overview of non-small cell lung cancer, the biology and issues at diagnosis. So you likely all know that lung cancer is actually the leading cause of cancer-related mortality in the U.S. And it accounts for approximately 1.4 million deaths per year worldwide and about 160,000 deaths per year in the U.S., which is approximately 25 to 30% of all cancer deaths and more than the next three cancers combined, which are colon, breast, and prostate. One of the reasons that lung cancer is often so difficult to treat at the time that it's diagnosed is that patients are often found at a very late stage, or stage 4. And at stage 4, this means that the tumor, which has originated in the lung, has actually metastasized and traveled from that original primary tumor, either through the lymph system or through the bloodstream, to other distant sites. And for lung cancer, the distant sites are often brain or internal organs, the adrenals, kidney, liver, or bone. And so often at diagnosis, people have already advanced to stage 4, which makes it very difficult to treat. There are two main subtypes of lung cancer: non-small cell lung cancer, with the abbreviation NSCLC, or small cell lung cancer. So today I'll be talking mainly about the non-small cell lung cancer because of the types of mutations it has relevant to the assay that we're using. So the non-small cell lung cancer can be further divided by the driver mutations that cause activation of signaling proteins that lead to tumorigenesis. And so I'll show you some examples and details about that in the next few slides. So the main mutation causing non-small cell lung cancer occur in the epidermal growth factor receptor gene. And I'm showing here on the side of this to the side of this slide, the EGFR protein, which is a transmembrane protein with a region on the outside of the cell that binds the EGFR ligand in this pocket, and then a part of the protein that exists within the cell and has many features that allow intracellular cell signaling for cell division. So mutations in the tyrosine kinase domain, which is shown here within the cell part of the protein. Mutations in this domain actually cause ligand-independent cell proliferation and survival. So this means that when you have a mutation in the tyrosine kinase domain, you don't necessarily need the ligand to bind to cause intracellular signaling, and this is what really leads to tumorigenesis when you acquire these mutations. And shown here to the side are just some examples of common mutations that occur in this tyrosine kinase domain. The tyrosine kinase domain is particularly important because it is actually the region where ATP binds to aid in the signaling. And so these mutations likely stabilize the binding or the association with ATP, and so produce this constitutive activation. We also know that these EGFR mutations are found in approximately 10 to 35% of all non-small cell lung cancers. And so it is a very common initial test on diagnosing a non-small cell lung cancer that patients will be tested for EGFR mutations. And testing for EGFR mutations then allows treatment with a class of drugs called tyrosine kinase inhibitors. Tyrosine kinase inhibitors, I show again here a little image, just a little schematic of the tyrosine kinase inhibitors, TKI. Tyrosine kinase inhibitors can compete with the ATP binding region here in the EGFR protein, and so by doing so, they help to prevent this constitutive activation of some of these mutations listed here. Okay, and just to look a little bit more at those mutations at the side that occur in the tyrosine kinase domain. And what I'm showing here in this slide is an overview of the EGFR, all of the exons laid out from 1 to 28, and then some of the important regions in the protein shown here, and specifically the tyrosine kinase domain, which is correlates with exons 18 to 21 of the gene. And so, as I mentioned, we know that there are EGFR mutations associated with sensitivity to TKI inhibitors, and some of these are much more common than others. So the most common mutations that give sensitivity to TKIs occur in exon 19. And this list here, this little triangle means delta or deletion. And so these mutations are actually all deletions within exon 19. And together they account for about 45% of mutations in EGFR in non-small cell lung cancer. The other very common mutation occurs here in exon 21, and it's called L858R, so that's a missense mutation, and this occurs in another about 40 to 45% of cases. So together, if you tested for exon 21 and exon 19 mutations, you would catch the vast majority of EGFR mutations causing non-small cell lung cancer. Some assays or approaches to testing for EGFR will also include other more rare mutations. So for example, there are some in exon 18 and 20. And so assays may actually try to capture those as well to increase the sensitivity. Okay, so in addition to these mutations that are associated with sensitivity to TKIs, we also know that cancerous cells can develop additional mutations associated with drug resistance. And this often happens after someone has been treated for a while with TKIs, that they'll develop resistance to the TKI drug and no longer respond to it. And that's often, again, due to new mutations, and some of those are shown here in the top in these yellow boxes. And these new mutations then basically prevent the TKI from acting in the way it was intended. And of these resistance mutations, the most common one is one called T790M, a substitution mutation, which is about 50% of the resistance mutations. So ever since they have developed the tyrosine kinase inhibitor as a drug treatment for non-small cell lung cancer and realized there was resistance, there was really a lot of interest to figure out how to identify these resistance mutations, and then what is the next drug treatment that we could use to overcome resistance. And so that was really why we undertook this study, that now there are new drugs that can actually work as a second line for people who have acquired resistance based on having T790M. So there was a need to be able to quickly identify people with that mutation. And so just sort of related to that idea then, recently the NCCN had written guidelines for non-small cell lung cancer, and saying that for individuals who had EGFR sensitizing mutations that had been treated and then acquired resistance, the new guideline really was to test them for EGFR resistance mutation T790M. So T790M can be detected through the sort of routine tissue biopsy where a sample would be taken, and a sample of the tumor taken, and then analyzed using molecular methods for that mutation. The real challenge there, and especially if you think back to those distant sites where lung cancer often will travel to, is just it can be very difficult to actually access the tissue. It may be in brain, which is really not something that can be accessed. It may be in bone, and so make it hard to extract DNA from molecular analysis, or embedded in internal organs. So there really are some challenges for people who are progressing about actually getting tumor tissue. But the other option then is what I'm going to talk mostly about then today, is using circulating cell-free tumor DNA. Circulating cell-free tumor DNA is DNA that is floating within your peripheral circulation, but originates from some cell somewhere else in your body. And circulating cell-free DNA, or cfDNA, can then really be isolated from just taking a regular peripheral blood sample, isolate the cell-free DNA, and try to identify any of that cell-free DNA that came from a tumor sample so that you can use that as a surrogate instead of actually getting a tumor tissue biopsy. And so just to help, with understanding what that really looks like, I've taken this picture that I think is very nice from a review. The reference is at the bottom if you'd like to have more information. So what's shown here in this picture then is just a sort of cross section as if it were through a blood vessel, and showing, arms leading off to capillaries to distant cellular sites. So if you think in this picture, the blue side is the tumor cells and the green is the healthy tissue. So if you think about in some distant site in a patient that they have a tumor that has developed, just naturally some of those cells will be dying or undergoing apoptosis during its life cycle. And during apoptosis of these tumor cells, many things are broken down from within the cell, including the nuclear DNA. This nuclear DNA can then be released and travel in circulation free of any cell envelope. And at the same time, healthy cells are also breaking down, and their cell-free DNA is also being released and traveling into the peripheral circulation. Or if there's any other inflammatory process or immune process going on, those cells will also be breaking down and DNA fragments ending up in circulation. And the thing, so certainly there is then obviously cell-free DNA in peripheral circulation. The other thing that is a bit of a challenge is that there are also cells within here, so the regular red blood cells or phagocytes from always in circulation. And in addition, there can be the rare circulating tumor cell. There has been some research for a while actually trying to isolate these cells, but it seems that they are so rare and at such low levels, it is very difficult to isolate the cells themselves, and a little bit easier then to actually try to get the cell-free DNA. So if you think about all of this stuff that is within peripheral circulation, then you can imagine if you took a sample, you'll get all of these things contained within your blood sample. And by spinning it and isolating only the plasma, which contain the parts in the liquid part of your peripheral circulation, and removing the cells, then you can capture the cell-free DNA. Okay. So in this way, as I mentioned, circulating tumor cell-free DNA is a really attractive approach to try to get a surrogate for testing or actually biopsying of solid tumors. It can be obviously taken at any time, and it can be taken in a repeated series if that is something that you need to, say, monitor or check for either early onset of a cancer or early recurrence of a cancer. And what does the DNA look like for cell-free DNA? And this picture just tries to explain what it actually looks like. So again, if you think about a person and in peripheral circulation, you have the breakdown products of different cells. The DNA, in the first one, it was a bit simple, just shown as a helix. But in reality, the DNA is wrapped around nucleosomes, and nucleosomes are packed into larger groups called chromatosomes, and which are shown here, chromatosomes in gray and nucleosomes in this pink. And so the DNA, as it's floating cell-free, is actually protected a little bit in this when it's looped around these protein complexes. And so, you know, within the peripheral circulation, the DNA not protected will be degraded, but still the DNA wrapped around the protein parts will be protected and saved and can be isolated. And so when you actually collect cell-free DNA and look at the fragment length of the DNA that you have obtained there from collecting in the plasma, you'll see two peaks, a really dominant peak at about 167 base pairs, which corresponds to the piece of DNA wrapped around the chromatosome, and then a more minor peak at about 174 base pairs that correlates to the nucleosomes. And so when you, if you're going to do next generation sequencing then, this size is actually quite amenable to producing a library of next generation sequencing, because typically we try to target right within that range of size. So the cell-free DNA does lend itself well to actually testing by next generation sequencing. Okay, so just to give you, again, some idea about our expertise with non-small cell lung cancer testing at Princess Margaret, we typically perform over 2,500 non-small cell lung cancer samples per year, because we're mainly looking for, at diagnosis, for the EGFR TKI sensitizing mutations. We have now, again, started to get into the resistance mutation testing. We do test tumors from all different types of collection methods. So from FFPE, from FNAs, corneal biopsies, or bronchial samples. We have several next generation sequencing panels for EGFR and additional genes, and also real-time PCR methods for specific mutations in EGFR. And so then starting in 2016, we did start to work up the NGS and also a digital droplet PCR method for the resistance mutation T790M. And so I'll show you a little comparison then that we did in this study between using NGS and using ddPCR for T790M. Okay, so just to sort of think a little bit about the pre-analytic phase, because a very important part about isolating cell-free DNA is that you're sort of optimizing the sample collection and trying to stabilize what you collect from a person's blood sample. The real risk with looking at cell-free DNA is that your lymphocytes in circulation are going to burst and then sort of contaminate your cell-free DNA with just normal DNA from the lymphocytes. And because the tumor cell-free DNA will be, you expect that to be quite rare in your circulation at very low levels. So you want to stabilize the white cells so they don't lyse, and you can really maximize collecting only cell-free DNA from distant sites that are in circulation. And so we actually use this a type of tube made really for collection of cell-free DNA called a Streck tube. It has a certain preservative within it that stabilizes the white cells. Once you put them in the Streck tube and with this preservative, they can be shipped at room temperature. So this makes it very easy for looking at studies or having to ship patient samples from distant sites. And then once the sample has been obtained, you spin it to isolate the plasma. So plasma obviously at the top, the white cells at the interface, and then red cells at the bottom, and then quantitate it. So for what we've, I often get asked how much blood do we need. So we take 20 mils of whole blood, so 2 of the Streck tubes, and that gives us about 8 to 10 mils of plasma. And from that plasma, we get on average about 80 nanograms of cfDNA. So it's not a ton of DNA, but again, if you have the right assay that can use a low level of DNA, you certainly can test it. Okay, so just a brief overview of next generation sequencing that we used in this cfDNA for lung cancer. So in this project, we used the next generation sequencing platform from Thermo Fisher, also known as the Ion Torrent system. For the platform from Thermo Fisher, you make a library by first either collecting or amplifying the small fragments that you want to test, and then connecting those onto these small beads where you amplify them in a PCR reaction. Each of these beads then should contain one original starting molecule amplified many times. And then each of those beads is put into a microwell plate, a special microwell plate that actually has on the bottom an ion-sensitive metal layer and then an electronic transistor type sensor. And the way that sequencing, next generation sequencing works in this system is that a solution of the nucleotides and other reagents like polymerase to extend the chain, copy the chain that is existing on this bead, is flowed across the cell, one nucleotide at a time. And so say you flow across a solution of A, and A is the next one to incorporate, it will incorporate, and that reaction will also release a hydrogen ion. And it's really this hydrogen ion that is the key part, because that hydrogen ion is detected by this metal sensing layer and produces a signal peak in the readout. Okay, so that's why if you've ever heard about the Thermo Fisher sequencing, it's really sequencing using ion detection. It's really using the ion released during the reaction of growing the chain embedded on the bead that is sensed and tells you what nucleotide is present. And so this is the kind of readout, if you could sort of see each of the sensing reactions going on as you float across the different nucleotides. So if you flow across T, that you would have a peak if that was incorporated, G, A, C, and so on. So you're sort of reading the sequence as it goes. And then if you have, say, two nucleotides adjacent to each other, so two T's, then the peak would be double what it would for a single one. Okay, so this then semiconductor type sequencing, and the real benefit for this, we thought, for using it for cell-free DNA, is that it doesn't take much DNA. For this assay, we only use 20 nanograms, and other assays that go on this machine also have a low DNA input amount. So that really does help when you're trying to work with cell-free DNA and you don't have much DNA to begin with. Okay, and then this slide shows an example of just the panels that are available, again, from Thermo Fisher on the same machine. So as I mentioned, I'll talk about this panel, the Oncomine Lung cfDNA assay, and it contains these genes, these 11 genes that are relevant to lung cancer. Again, for this study, we're mostly interested in EGFR because we wanted to look at T790M, but it certainly does have other genes relevant to lung cancer. Not all of these genes are fully covered. It's more of a hotspot panel looking at known single nucleotide variants and insertion deletion mutations. They do also sell a colon cfDNA panel with 14 genes, and then a breast cfDNA panel with 10 genes. So again, each of these are sort of optimized for those particular disease sites. Because of the need to test sort of a low level of DNA and to have a very high amount of sequencing done to have a very sensitive assay, it makes, I think, more logical sense to have somewhat smaller panels specific to disease sites rather than a pan-cancer large panel, which may have too many genes to really be cost effective for the type of sequencing you have to do. So I think that has been the approach here. And I also wanted to mention that the limit of detection is, again, very key for cell-free DNA, that we really wanted to be able to achieve a very low level, and here they're sort of promoting it as a 0.1 limit of detection, because that, again, for cell-free DNA, we wanted to be sure if we said it was negative, that we felt pretty confident that we weren't just missing it if it was at a low level. And again, just a little bit of an overview about the assay then. We're using a very small amount of input DNA, 20 nanograms, trying to get to a 0.1% limit of detection. We did run it on the S5. Thermo Fisher has several different models. We ran the S5 system, and those are the genes and the hotspots covered. And the hotspots, of course, then included T790M. And this shows you the workflow then this is specific for this Oncomine CF assay, but really it's the workflow for almost any NGS, next generation sequencing assay. So obviously you started with some kind of DNA isolation. Then the next step after you have your DNA is really to do the library prep, where you're trying to get those fragments, put adapters on the end, and get them ready for the actual sequencing. For the Oncomine cfDNA, we again use 20 nanograms of DNA. Then the next step is to actually set it up and do the sequencing. The Thermo Fisher system does come with some automation called the Ion Chef that helps you do the setup for the actual setup of the library and for the sequencing reaction. So that really does help with throughput. And then after that comes the analysis. So the analysis is the part where you're taking all of the reads that you had generated in your sequencing, trying to align them to reference, trying to link them back to any known databases to see what mutations they are, and then also trying to analyze quality measures such as are there any artifacts, and what is the lower limit of detection of your assay. And then we generate, if it's going to reporting, we would generate some kind of report. This is showing using the Oncomine database, which Thermo Fisher sells, but there are many other databases which can be used to help generate your report and interpret the data. Okay, so just a few words then about the lower limit of detection. So we know that next generation sequencing has artifacts that appear as variants below 5% allele frequency in the results. And this just has to do either with technical methods that cause a low level of artifacts, or something to do with the actual informatics, the aligning of your data to the reference that causes misalignment and problems. And so as I had sort of alluded to, it's very important to have a really sensitive test for cfDNA testing. And this is showing a picture actually of testing from, say, a solid tumor sample, but I think it just illustrates the point that I want to make, that if you think about having a solid tumor and you take a sample from it for sequencing, sometimes within that tumor there might be different clones. So you might have one clone like this blue one with a certain mutation, and then a red one that has that mutation but it's acquired a new mutation. And so this mutation that is the white block would be expected to be at very low levels compared to other mutations in there. So if you think about all of the reads that you would get from your next generation sequencing, sometimes you would have changes in those reads that would correspond to common mutations. So they may be in every single read. They may be either a really high occurrence mutation or even something like a germline polymorphism. And then other times you'll get other changes that are much more rare. So here you can see a C, and here an A, and here 2 T's. And the challenge really is to differentiate, is this C caused just by a sequencing error, or is it caused by a specific clone, subspecific clone mutation? And it's the exact same thing in cell-free DNA, that you still get errors that come up when you isolate cell-free DNA, and you're trying to figure out which of these errors are real and which of them, which of these small frequency variants are real and which of these are actually just sequencing errors. And so the way that this has been done with the Oncomine CF assay is they have sort of inserted what they call a molecular tag. And this molecular tag is a way that they are using to help suppress the possibility that a variant you identify is actually an error rather than a real mutation. And the way that this works is, as the assay is set up to amplify the region of interest, they include within the primer set molecular tags. And the molecular tag is, one for the forward and one for the reverse primer. And what that means is then when you have a region that contains a variant, and these are showing that the 2 strands of the region that you amplified, each of these strands from that same molecule will have the same molecular barcodes attached to them. And the same is true for wild-type DNA. They'll have codes, a different code attached to them too. And so then when you do your actual sequencing reaction and combine all the reads, this variant may be at very low levels in the overall group of reads that you get. But when you identify it, you can then sort of work backwards to say, whenever I see this variant, do I also see the same molecular tags on the ends of that read? And if the answer is yes, then you can be much more confident that that variant is a real variant, because you would expect if it was just a random error, it could appear sometimes in this barcode, sometimes with that barcode, so it would just occur much more randomly across the set. Okay. Okay, so then this assay, when they first set it up, they had publicized the lower limit of detection and how they actually could use this assay at a very low level for many different mutations, including EGFR mutations. And this slide is just showing you a study they did using a reference material from Horizon that, excuse me, that contained these 8 different mutations across a few different genes, and then diluting them down to different sensitivity levels. And what they showed in this study, and this was a worldwide study, and what they really showed in this study then was that the assay was very good at picking up down to 0.1% limit of detection for these variants. And of course then it was also quite reliable and reproducible at 1% and 5%. So then, again, just sort of back to our study. So in our study, we did use these Streck tubes that I mentioned that do stabilize the nucleated blood cells and prevent release of the genomic DNA. They are stable for up to 14 days at room temperature, and really does help to reduce the need for immediately isolating the plasma from after collection, as you would have to do if you didn't have this stabilizer. And so in our study then, we had taken samples from 62 non-small cell lung cancer patients and put them in the Streck cell-free DNA blood collection tubes. And we had obtained about 8 to 12 mils of plasma from the blood for each person. And we extracted using the Patien circulating nucleic acid kit from QIAGEN. The concentration of cell-free DNA that we obtained was about 0.2 to 26 nanograms per microliter, with the total DNA amount being a median of 80 and anywhere from 8 nanograms to 1.6 micrograms. Okay, so I did also mention that we used a comparison assay to look at the results to compare the results of our next generation sequencing assay and we had used droplet digital PCR. So droplet digital PCR, I just gave a little overview in this slide. A droplet digital PCR, if you think about a sort of conventional PCR, if you had in a tube many normal molecules and one mutant molecule that you wanted to identify, it would be very hard because as you amplify in the entire tube, your mutant signal in green here would be sort of swamped out by the wild type in red. And so it can be very difficult to have a really sensitive limit of detection with routine, regular PCR. So instead what happens in droplet digital PCR is that your starting material is sort of encapsulated into these droplets shown here. And each of these droplets becomes like its own small miniature PCR reaction. And, you know, of course, then most of them will have the wild type, but by chance, some of the droplets will contain the mutant that you're trying to target. And then you carry out an amplification within these droplets, and the results of the droplets are read by the machine, and using statistics, it's calculated really what was the percent of mutant and wild type in your original starting sample. And this is showing the type of data, the sort of raw data that you get from a digital PCR assay. The 4 quadrants here are marked with whether the actual, as the machine is reading each of those droplets, it's classifying it as to whether it is a mutant droplet appearing in blue in the upper quadrant, wild type droplet, which is green in the lower quadrant, or by chance some of the droplets will contain more than one molecule, and so those could be classified as wild type and mutant in the upper right quadrant, and then the black are just empty droplets. And so just by looking at these 4 different graphs that I show here and the percent mutant above them, you can tell that it is pretty easy to quantify the ones that actually have a much higher percentage of mutants from those that have a low percentage. But it does allow you to get down to a very sensitive level of detection. So here we're showing 0.8% mutant, a case of 0.8% of the mutant of this particular variant. And we typically are able to validate the different digital droplet PCR assays down to 0.5%, possibly even lower depending on the variant. And so we felt this was a good comparator. We did have a ddPCR assay for T790M and EGFR. And so we felt this was a good comparator to be able to compare detection using NGS with ddPCR. And so when we first got the CF oncomine lung assay, we did do the same thing that the worldwide study had done, which was to take the Horizon reference control and look at all the variants and could we get down to the level that they said. Generally, yes. We did have a problem here with one of the mutations, one of the deletion mutations in EGFR. We couldn't detect at 0.1%, and we could, however, find it at 1%. And again, I had mentioned that we knew from ddPCR our limit of detection was really 0.5%, that 0.1% was not always reliable. So we weren't too concerned about that. It did seem to be at least comparable to our ddPCR assay. And then, so just again, we had just an overview of the study. We had done 63 samples on both ddPCR and then the Oncomine lung. And just to reiterate that ddPCR, we really were only looking at one mutation because it just does a single mutation at a time for the singleplex reaction. And then for the Oncomine lung, we were looking at the 11 genes. DNA input was very similar, and sensitivity, again, similar, 0.5 to 0.1%. And then below, I'm just showing sort of the general results of the study. So of those 63 samples, for ddPCR, 49 passed and 12 failed with 2 insufficient. And for the NGS Oncomine, 60 passed, 1 failed, and 1 was insufficient. So the first thing that might jump out is that we had a pretty high fail rate on the ddPCR. We weren't expecting that. We had used ddPCR on many other types of tissue, not cell-free DNA, before this study. And so that was something that was new to us, that cell-free DNA actually did not work well in ddPCR. This slide is just showing you an overview of some of the different runs that we did to give you an idea of the quality metrics. Generally, all of the quality metrics for the runs were very good. They did vary a bit, and probably partly as we gained our expertise. So here, for example, the goal here in this column is to have greater than 50. Not all of them met it, but once we got more familiar, really the quality metrics were good, and they gave a good amount of sequencing as we would expect. And then the same, this is actually looking at the quality metrics for each sample. Most of the vast majority of the samples gave great results on the assay. We did have a couple samples I highlighted in red at the bottom, one of which was suboptimal, and then one of which really failed on the NGS Oncomine assay. So just to mention a bit about this, the samples that failed the ddPCR, we did want to see, just look at those ones because they did work on the NGS assay. So, would we have missed anything if we weren't able, if they failed because of the ddPCR? And so the answer to that was definitely yes, that of 11 samples that we could retest that had failed ddPCR, 4 of them actually had the EGFR T790M variant at a relatively high frequency. And so, so that really made us much more interested in using this assay for T790M rather than ddPCR. And then this is, again, a comparison of all the samples that actually worked on both assays, so both ddPCR and the CF Oncomine lung assay. So of those 49 samples that worked on both assays, the positive at greater than 0.5% was the same for both ddPCR and the NGS Oncomine at 22. 2 of them had a low positive between 0.1 and 0.5%, and that was the same in the Oncomine lung assay. And 25 were negative for ddPCR. However, on the Oncomine lung assay, we had only 15 that were negative. And the difference here is that we actually had 9 samples that were negative on the ddPCR assay, but positive on the lung cfDNA assay. So we would have ideally liked to go back and reconfirm that that was sort of a correct test from the Oncomine lung assay. But unfortunately, we didn't really have much DNA left over just because of the problem of collecting a small amount of cell-free DNA. So for further studies, we're trying to collect more DNA so that if we ever have this kind of something we want to confirm to say, yes, this was better at detecting these variants, we can do that with more DNA. And overall then, the variants that we found really had a range, and that's shown here, had a range anywhere, from very low, 0.1%, all the way up to 50%. And this graph is just showing comparison of the allele frequency of the NGS Oncomine assay versus the ddPCR on this axis. And so correlation was very good between the variant allele frequency found on both assays. And so that really is about our T790M variant. But I also put in this picture just to remind you that, of course, the NGS assay has many additional genes that it tests. And so just to show you, in some of the cases where we also looked at these other genes, we could definitely detect them, and again, down to very low percent. It was quite easy to do at 5% and 1%, but could also get down to 0.1% for some of these other variants. And some of the other variants include things like the EGFR sensitizing mutation. And so that may actually be to test both the sensitizing mutation and the resistance mutation. That can also maybe be a very helpful way just to confirm that you have circulating cell-free DNA from the tumor within your sample. I talked a lot about the lower limit of detection and how important that is, and this assay, for people who are interested, does also give some metrics to allow you to try to optimize your ability to detect down to 0.1%. That would certainly mean depth, so how many reads you have at a specific location is important, and the metric is shown here. It should be greater than 20,000. Mean molecular coverage greater than 1,500, and then targets at that greater than 80%. And so this was pretty easy for us to achieve. Sort of 85 to 97% of our samples did meet these coverage metrics, these quality metrics to achieve this kind of sensitivity. So we were happy with that. And so overall then, based on the NGS Oncomine assay, we had 37 cases that were positive for T790M with this allele frequency and 23 negative. And again, we could also find that in the ones that had T790M, 36 of these, the vast majority, had also one of the sensitizing mutations, because again, these were patients who originally had a sensitizing mutation and were then treated and developed resistance by T790M. And just a little overview of the other, these are the the EGFR mutations we also identified in those patients, as I mentioned, and then some of the other variants and other genes that we found in these cases. So 10 of the cases had TP53, which is also very common in non-small cell lung cancer, and RAS and PIK3CA. So yeah, and finally, just my final slide, just to kind of remind you the overview of this whole assay that we tried. A small amount of input DNA, looking at multiple genes, and particularly trying to assess the utility of using it for detecting T790M for resistance in EGFR for non-small cell lung cancer. And finally, just a thank you to all of the staff who at our labs and the group who has worked with this. And a thank you to Michelle for suggesting this as a topic for your seminars. Thank you.
So thank you, Dr. Stockley, for an informative session. So I do hope we may have some time for a bit of questions. So let's see if we have anything that's queued up here.
Okay. Sorry, I can read this. Yes, sorry. The question is, do you validate by comparison with non-malignant tissue from the same patient, or do you pool DNA from 20 unrelated individuals? Yeah, we certainly did run some normals as we validate. In this particular assay, I think it's maybe not quite so relevant. We were just really trying to use this on tissues that already had known mutations. I think the question there is maybe more about if you're doing tumor normal match testing, where you are looking first at a tumor and trying to see if the variants within that tumor are important because they're not also in the germline. Here it was a little bit different. These were tumors in patients who already had a known diagnosis of non-small cell lung cancer and already had EGFR mutations identified. So what we were trying to do is really trying to see if the known mutation, T790M, actually occurred in cell-free DNA.
Do we have any more? Let's see. And as a reminder, you can type your questions in the webinar's control panel.
Okay, so, oh yes, so the next question is, would you consider using an NGS panel using the Illumina technology? Yes, definitely, and yeah, I definitely want to be platform agnostic. We do that as well. We do cell-free DNA we have the Illumina platforms here, and we definitely have cfDNA panels on Illumina. It works very well as well. For those, Illumina doesn't produce custom commercial off-the-shelf panels, so for those we do custom panels. Yes. So definitely important, a very important question, yes. Okay, sorry, I'm just trying to scroll through and see if I can see any others. Oh. What about nanopore? Yes, it's very, very trendy, I know. I haven't tried nanopore myself, so yes, I'm not sure. I think, you know, just the theory of it, being able to actually do long reads using nanopore is very attractive, especially for things like structural rearrangements, for sure. But here at Toronto, we haven't yet tried it, yes. Okay, maybe that's all. Yeah.
Well, I hope we have time. Actually, I had a question because I wasn't working on this project, Dr. Stockley, and I was wondering, it was that slide you had about the pre-analytical setup, and
right.
It was a very nice slide, and I understand that for cell-free DNA, that can be a challenge due to the variable shelf life, especially if you're looking for the tumor DNA. So were there any constraints in that setup for pre-analytical, from, you know, collecting samples from patients at different time points to even how to measure cell-free DNA in the lab?
Yeah.
Were there?
Yes, definitely. And in fact, that was part of the goal of this study, was to actually say, can groups from across Canada send us samples and we get apparently consistent amounts of DNA. So that was a huge part of it, because even at the beginning, you know, we had samples shipped from, you know, a 2-day shipping time to us. That was a huge part of it. So that part where I showed about amount of DNA and the range and all of that was a very critical thing to be able to see if that actually worked. There is a lot, you know, I do think those cell tubes really do help a lot, but there is a lot we don't know about how much samples change really as soon as you take them. It does stabilize. We know it stabilizes the white cells and should help with that problem. But yeah, there is, I think, still a lot of unknowns about the pre-analytic for cell-free DNA for sure. So we try to control it as much as we could with those tubes, yes.
If we have any more queued up questions. If we don't, I actually have one more. Okay, and I think, all right, I have one more question. So I know for ddPCR, when we were comparing this in the lab here, that's a very quick turnaround test. And for cfDNA, that's probably half a week that we're looking at. So how is the lab, how do we move forward with, you know, do we balance sensitivity with turnaround time? I'm just not.
Yes, that's an excellent question, and you know, we really, I think at this point, still don't have answers to that. Absolutely, the ddPCR is a really good and simple test and very fast. So I mean, it may be, and our approach really, when we're moving it over the clinical side, has actually been that we actually need both available. That we need ddPCR for some very simple assays that fast turnaround time and quick, and then maybe we need NGS for other patients where we're looking a bit wider at the genes, or something more complex is going on. So definitely there is always that trade-off of, really for anything you're comparing to NGS, of the time.
Thank you. So I think that's all the questions that we have, and I want to thank you all, Dr. Stockley especially, for presenting, and for everyone for participating in this webinar. Shortly, the webinar participants will receive an email from AGT that includes a link to an evaluation. Please take a few minutes to access it and provide your feedback on today's session. The webinar today is recorded and will be posted on the AGT website in the near future, and we hope you will join us for upcoming webinars over the next few months. We will send out a notification to members once registration is available for our next seminar, and we hope to see you all at AGT's 2018 annual meeting in Tampa, Florida. Yes. Well, thank you all.